Hello. Thank you very much, Dr. Bridget, for this great opportunity. It's a great pleasure to be here with all of you today. This event is a very special one, and it is a great pleasure to be representing Brazil as my lecture. I will share my screen and start my presentation. Sorry. In order to avoid compromise my presentation with my picture, I will close my video, and so we can go ahead. So the title of my presentation today is Antioxidant Effects of the Oral Use of Propolis Extract, a before and after clinical trial. For whom don't know me, I am Andresa Berretta. I am pharmacist. I have my master and doctor and postdoctoral degree at University of São Paulo in the Faculty of Pharmaceutical Science from Ribeirão Preto. I am currently the head of R&D at Apitherapy, a bee products company, and I am currently the president of ABML. I am now the convener of Working Group 2 at ISO in the leadership of Propolis standard elaboration. And also I am a member of ABNT, the standardization body at Brazil. This is my R&D team at Apsflora, and our results will be presented in the next slides. I would like to start my presentation talk something very fast about oxidative stress, since it is very important in the aging process, or in the case of the disease that we will have with the aging. Maintaining cell viability requires a continuous process of energy production, which is largely accomplished by mitochondria. In this process, reactive oxygen species are formed, many of which participate in the control of cellular homeostasis, acting as biochemical intracellular signaling or even controlling gene expression. Under condition of hypoxia, aging, or cell damage, this respiratory chain may be decoupled, leading to the formation of reactive substances such as aldehydes, malondihyde, superoxides, and peroxides, which in turn damages nucleic acids and cellular membrane. Under physiological condition, there is a balance between free radical production and its elimination. However, excess of production of oxygen-free radicals, defined as oxidative stress, can lead to cell damage and be part of a number of some chronic degenerative diseases observed in humans, such as cancer, inflammatory and cardiovascular disease, diabetes, atherosclerosis, and Alzheimer's, besides some others. Antioxidant defenses are composed of enzymatic mechanisms such as glutathione peroxidase, catalase, and superoxide dismutase, and dozens of substances that play a role in binding and neutralizing free radicals. These substances may be endogenous or exogenous, as some food supplements as flavonoids, carotenoids, polyphenols, ascorbic acid, and vitamin E, besides others. In our case, the complementary medicine can be from bees as royal jelly, propolis, honey, and others. Considering some chronic diseases have pathophysiological mechanisms involving oxidative damage by free radical generation, there is an interest in studying the use of functional food or nutritional supplements with antioxidant properties to prevent the worsening of such chronic diseases. Thus, the main objective of this study was to evaluate the antioxidant effect of standardized propolis extract for oral use in healthy volunteers. I would like to start with a preclinical study that we did in vitro model, in fact, besides to introduce the clinical study. This study was published some years ago and was entitled Effect of Brazilian Green Propolis on the Production of Reactive Oxygen Species by Stimulated Neutrophils. So, the purpose of this study was to evaluate the percentage of the inhibition of some extracts as alcoholic extracts and some fractions as butanolic, chloroformic, and oxidant fraction. And we used the quercetin as the standard. We used the chemiluminescence as the model, and the neutrophils was stimulated with zimosan in order to produce the free radicals. We used some models as luminol and lucigenin to evaluate the percentage of inhibition of the fractions used, and quercetin was the reference standard. Here you can see that the reference standard is this curve of inhibition is here, and we have several others. And it's possible to see that the butanolic fraction and the chloroformic fraction obtained the best antioxidant activity in this first model, followed by the others. The alcoholic extract is here. In the case of the food additives, food supplements, we usually use the ethanol extract of propolis. But here we only fractionated using some solvents in order to try to identify the compounds that were involved in this process. In the next stage, I will show you the chromatograms. In this other protocol, we also observed the antioxidant activity in this second model with some more similar results between the curves, considering the statistical analysis, different from this one, where butanolic and chloroformic fraction obtained the best results. And the isolated compounds that we studied was camphoride, isosaccharinetin, aromatendrin 4-methyl ether, triprenyl paracumaric acid, and quercetin, that was the reference standard. And the results of this evaluation demonstrated that camphoride was so potent as quercetin, followed by the others that was very close in the antioxidant activity. Here, the isolated compounds that we evaluated, demonstrating that camphoride, this flavonoid, was the most important one. Here, the fingerprint of the fractions of butanolic fraction and chloroformic fraction, demonstrating that the compounds identified here, and some of them was isolated and evaluated in this model. Camphoride is this one. Here, the results of the percentage of the inhibition, demonstrating that the butanolic and chloroformic fraction demonstrated the highest percentage of inhibition, followed by camphoride. And in this other model, the cytotoxicity assay was done in order to check if the antioxidant action or the result was involved with the death of the cells. That was not the case. Finally, we can start the clinical study. This clinical study was already published in 2020 and was the title of this presentation. This study was done at the University of São Paulo Hospital, and we used here as the medicine the green propolis extract standardized. This green propolis extract was prepared from a blend of Brazilian different types of propolis, and we previously demonstrated the reproducibility batch to batch in order to have the standardization. We have already approved this product with a patent letter. This protocol have as the outcomes, as primary outcome was to evaluate the antioxidant efficacy of EPPAF using the urinary excretion of 8-isoprostanes, a marker of in vivo lipid peroxidation, and 8-OHDG, that is 8-hydroxydeoxyguanosine, a marker of oxidative DNA damage, as surrogate biomarkers. This was the first publication that we saw that used this biomarker of oxidative DNA. The secondary outcome was the safety of EPPAF in healthy volunteers assessed by kidney and liver biochemical tests. The exploratory outcomes were focused on studying some biochemical pathways involving oxidative stress. we studied the ferric reducing antioxidant power, FRAP, vitamin E superoxide dismutase activity, and GSSG and GSG in plasma. That is the reduced glutathione and oxidized glutathione. Also, the malondialdehyde assay was done to detect lipid peroxidation in plasma. In this protocol, we did a treatment that involved, in the beginning, we collected the blood and urine sample, and after this first collection, we treated the patients with EPPAF with the dosage of 350 milligrams per day by oral route. After 7 days of treatment, another collection of blood and urine sample was done in order to evaluate the effects in plasma and urine as previously said. And so the next 7 days was done treatment with 700 milligrams per day of EPPAF. This clinical trial was 2-phase sequential study, open label, non-randomized, before and after clinical trial, because we evaluated the same group of patients before starting the treatment and after the treatment with properties EPPAF. So the same group was the same treated and its own control. In this protocol, the values was considered, afterwards, the participants were instructed to use, okay, this was the protocol that we ran. Okay, the inclusion criteria were to be healthy participants, no history of allergies, no continuous use of medication 1 week prior to the study, and having read and signed the informed consent form. With, in the case of exclusion criterion, we excluded pregnant women, women breastfeeding, smokers, participants with history of alcohol and drug abuse. So the treatment, this protocol to do this clinical trial, we assessed 56 patients, and from these 19 were excluded, some because of non-meeting inclusion criterion, and some because declined to be part of the study. We allocated in the study 37 patients that received the medication. The follow-up was done, and we have 3 patients that discontinued the intervention, one that lost the follow-up visit and declined to be part of the study, and some that stopped the intervention by adverse events, one because headache and another one because of diarrhea. And in the end, we have in the end 34 patients that were evaluated. Here you can see the biochemical safety profile before the baseline and 30 days after the end of the EPPAF treatment with the highest dose. The biochemical tests done 30 days after the last dose did not show any hematologic metabolic changes or either kidney or hepatic damage compared with baseline. In addition, the frequency of adverse events was low, only 3 cases. That corresponds to 80%. All mild and potentially unrelated EPPAF use. In this context, the present work shows that EPPAF at doses up to 750 milligrams per day was safe and does not promote oxidative or toxic effects, at least in healthy humans. Okay, so in the next in this table, we can see the effects of the results of the biomarkers evaluated. In the case of FRAP, values showed no differences after the use of both doses tested. Similarly, there was no effect of EPPAF on plasma vitamin E concentration. There was a significant elevation in plasma GSS concentration in both only in the low dosage compared with the baseline. SOD activity also increased compared to the baseline in the both dosages. There were no significant changes in the amount of GSS in the periods before and after the use of EPPAF, as well as in the GSSG ratio. Superoxide dismutase activity was expressed as a percentage of inhibition and increased significantly with the use of EPPAF from baseline. The maximum effect observed was with the low dose. Superoxide dismutase activity was correlated with urine 8-isoprostane values. Here in this graphic, you can see both dosages different from the baseline and the correlation between superoxide dismutase and 8-isoprostane. There was no statistically significant correlation between GSH concentration and oxidative stress damage markers. Here we can see there was a significant reduction in urinary extraction of 8-isoprostane with the use of EPPAF. Apparently, the maximum effect was observed at the low dose, since increasing the dose was not accompanied by an increase in the magnitude of the effect. There was a reduction in urinary expression of 8-OH-GG only in the higher dosage. There was no change in plasma malondiide values. So it is possible to see that we have important effects in 8-isoprostane and in the biomarker of DNA oxidation process. This apparent contradiction could be explained by the lack of sensitivity of the in vitro MDA method. This result of malondiide, that is apparent contradiction, probably is linked to the sensitivity of the in vitro method compared with the measurement of 8-isoprostanes, which are chemically stable markers and are formed in vivo. They are specific to lipid peroxidation and are not affected by the dietary lipid potent in contrast with MDA. In the end of this study, what we can conclude is that EPPAF has an important antioxidant effect in humans. This effect does not involve consumption of endogenous antioxidant substances as vitamin E or glutathione system. There is a reduction of cell membrane damage and DNA damage. Most of the antioxidant effects was observed at a dosage of 375 milligrams per day of EPPAF. The reduction of the 8-OH-GG biomarker of DNA damage was observed only at the higher dosage. Part of this phenomenon may be associated with the short treatment time, only 7 days, and low harm potential of participants, young and healthy. This is something very important. We only tested in healthy volunteers, not in patients with inflammatory process. Because of it, maybe this could impact in the malondiide result. This study possessed some limitations. The main limitations of this study are in its design. As it was a proof of concept study, a before and after design was chosen. There were no blinding, randomization, or placebo control, which may inflate the magnitude of the observed effect. Also, doses were elevated without a washout period, and a cumulative dose effect may have occurred. The safety concerns of EPPAF use could not be generalized, since we only study a small group of young humans without underlying disease or concomitant use of drugs. Despite these limitations, the study's strength are to use healthy young people at different dosage exposures with a set of biomarkers being considered as the gold standard for detecting oxidative stress injury in humans and performed with a standardized presentation form of properties. In conclusion, the present study shows for the first time that a standardized properties extract led to a reduction in urinary excretion of 8 isoprostanes, which has been correlated with a rate of lipid peroxidation, and reduction in OHGG, which has been shown to be a reliable marker of oxidative DNA damage in humans, associated with increase in enzymatic protection capacity for redox damage. To see one application of this study, because we studied in healthy volunteers, I found in the literature, there's another study that is able to show an application in a group of elderly Japanese. So this work was entitled Cognitive Improvement and Safety Assessment of a Dietary Supplement Containing Properties Extract in Elderly Japanese, a placebo-controlled randomized parallel group double-blind human clinical study. As we demonstrated the impacting effect of antioxidant action of propolis, here in this work we could see the important effect in the composite memory and verbal memory, where after 24 weeks of treatment, the group that received propolis, that is this one, demonstrated after 24 weeks a significant improvement in this effect. And in the case of verbal memory, too, we have an improve. In the case, a significant improve in relation to the placebo group, in the case of propolis, demonstrating that the antioxidant effects of propolis can be very useful in the elderly, in the case of the degenerative disease. So this was the most important result of this study, demonstrating that propolis can help in the elimination of some important effects of, in this case, of degenerative disease. To conclude this presentation, I would like to celebrate with you the 40 years of Apitherapy, the company that I am doing my research during the last 20 years. The company are completing 40 years in 2022. And we would like to celebrate with our thanks to all of our partners that involve partnerships in the Brazil, as USP, Unicamp, Fiocruz, Unesp, University of Maranhão, University of Paraíba, University of Brasília, and several others, and the international partners, as Instituto Politécnico of Bragança, Chubu University, Jumonge University, and some universities in France, and some others. Apis Flora is present in more than 40 years, is present in more than 40 countries, is now a company member of Lenin Group, and is the first company to produce and commercialize propolis extract in Brazil. We have more than 15 certifications. According to the IQVIA, Apis Flora is the company leader in propolis extract sales in Brazil in the year of 2020. This year, to celebrate our 40 years, Apis Flora launched in the market a kids line. This kids line uses a technology of eye caps that involved the microencapsulation of propolis extract in order to avoid or in order to mask the taste, the flavor, and the smell of propolis, but increasing the absorption of propolis in the organism and have the same effects that we usually have in propolis as antimicrobial, antioxidant, and some other effects. This publication about the microcapsules are under application. And also, we introduced 2 years ago the line in the French market, which involve the same same products that we have in Brazil, but with a special brand to be commercialized in France and in some countries in Europe, as Portugal, Spain, and others. We did a very important celebration with more than 80 partners from research that we have in order to present our results of our research. We have here the founders of the company, Mr. Antonio Carlos, Mr. Manuel, and now they are retired, but their sons, Raul Ferreira is our CEO. And here we have the event of the introduction of new products in the market. Here's some of our research partners showing their results on their poster presentation and also as speaker in some presentation. We have 2 days of this very impacting event for us. Also, this newspaper in Brazil called Folha de São Paulo is a very important one, and present the result of an important agency of research in Brazil. This agency called the FAPESP is very important in supplying funds, funds for research, not only in universities, but also in companies. They did evaluation of the companies that are publishing scientific results. And surprisingly, Apitherapy was cited in this research as a very important company in the publications in partnership with universities. They ranked the publications and the companies that were doing research with the universities, and Apitherapy was in the 23rd place, in front of some very big companies as Sanofi, Microsoft, that was with less publications than us. So this was a very impacting information that we celebrated with our results. We are doing research with some important agencies in Brazil, as FAPESP, that is from the São Paulo state, CNPq, that is a federal agency in Brazil that supports research, and FINEP, that also supply funds for research. Thank you very much. I hope you enjoyed the content of this presentation, and I believe, I hope you have the opportunity to do a contact if you need some additional information. So thank you very much. Thank you for the organizers, and congratulations for this big, great event. And today in Brazil is yet November 19th, my birthday, and it's a great pleasure to celebrate my birthday with you, showing one part of our results. Thank you very much.